gidiyor.
Şimdi geç hocam.
Geldi mi ses? Diğer tarafa. Geldi. Alright. There are, as you have learned, I'm not going to give you lots of theoretical information, but there are primary and secondary lymphoid organs. What were the primary lymphoid organs? So there were bone marrow and thymus. And secondary lymphoid organs like lymph nodes, spleen, and MALT, mucosa-associated lymphatic tissues. All right. So in this laboratory, I'm going to show you thymus for a start. So this is the lobus magnification. This is the lobus magnification of the thymus. And as you can see, the thymus is surrounded by a cortex, okay? So this is surrounded by a cortex. So this cortex extends into the organ. This cortex extends into the organ, as you can see. And these are called septa. So this cortex extends into the organ and gives rise to septa. And these are all blood vessels. These are all blood vessels which brings the lymphocytes to this organ. And in this lobus magnification, you can see the parenchyma. This is the parenchyma of the organ. The parenchyma of the organ is divided into cortex, this dark stained area. These are all cortex, okay? This is all cortex. And this is the light stained area is the medullary region of the organ, okay? So this is the capsule of the organ, and this capsule extends into the organ and gives rise to septa. So these are all septa. And the parenchyma of the organ is made up from cortex and medulla. And as you can see, the cortex is outside of the medulla and stained dark. This is the medulla, cortex, sorry. And this is the medullary region of the thymus. And as you know from your theoretical lectures, the cortex of the thymus consists of lots of lymphocytes, T lymphocytes, lots of T lymphocytes. And these T lymphocytes, they are trying to differentiate, they are trying to become mature, all right? So in the cortex of the organ, you will see lots of T cells, T lymphocytes. But as you know from your theoretical lectures, without using antibodies, it's not possible to differentiate which type of lymphocytes are these. But all we have to know is in the cortex of the thymus, there are lymphocytes in differentiating stages. And when we look at the medulla, in the medullary region of the thymus, you will see lots of macrophages, you will see lots of dendritic cells, but few T lymphocytes. You will also see medullary cords and sinuses. So let's magnify our image. Okay, so this was my capsule. And capsule gives rise to septa. And in the parenchyma of the organ, we have cortex, this dark stain. See, all this area is cortex, okay? These are all cortex. And this is the light stained area is medulla. And in the cortex, as I have told you, you will find lots of lymphocytes, T lymphocytes. And in the medulla, you will see lots of macrophages, dendritic cells, epithelial cells, and sinusoids. And as a feature of thymus, I'm showing you the Hassel's corpuscle. Hassel's corpuscle. You know this from your theoretical lectures. When epithelial cells, let me try to show you. So I'm in the medullary region. I'm in the medullary region of the thymus. I'm in the medullary inside the lightest area of the thymus. And in this area, as I have told you, you will see macrophages, dendritic cells, epithelial cells, but also T lymphocytes. But the number of T lymphocytes in the medullary region is much more lower than in the cortex region. And epithelial cells, you know what are they doing in your theoretical lectures. See, this is an epithelial cell, okay? You may see individual epithelial cells, but sometimes these epithelial cells, the whole of epithelial cells, the concentrated epithelial cells, comes together to form Hassel's corpuscle. So this is a Hassel's corpuscle. Hassel's corpuscle means the concentrated epithelial cells. See, I'm showing you an individual epithelial cell. When they come together and concentrate into each other and make a whole of epithelial cells to form Hassel's corpuscle. Okay? So in this first slide, all you have to know is thymus is made up of, thymus's parenchyma is made up of, what is it? Cortex and medulla. In the cortex, you will see lots of T cells, and in the medulla, you will see macrophages, dendritic cells, but less T cells. And you should also try to find Hassel's corpuscle. Hassel's corpuscle, which are concentrated epithelial cells. Okay? And let's go to the second slide. And the second slide is an example of lymph node. It's an example of lymph node. See, this is the lowest magnification. This is the lowest magnification of lymph node. And in the lecture that we've done together, I actually mentioned lymph nodes. So we have lymph in our body, which are the extracellular fluid in our tissues, and all this extracellular fluid is being collected by lymph vessels, yeah? And these lymph vessels brought all this lymph to lymph nodes to be able to filter the lymph. So this is our filter, okay, the lymph node. I'm showing you the filter of our body. So this is the, I'm sorry. So this is the capsule. This is the capsule of the organ. This is the capsule of the organ. And as you can see, there are lots of vessels here. There are lots of vessels here. And I can see lymphatic vessels. See, this is the lymphatic vessel. And this structure is the valves. Remember, all lymphatics have valves. And I can see the valves of the lymphatics. Another lymphatic blood vessel. And this is the valve of the blood lymphatic vessel. So all the lymph enters to the organ, all the lymph enters to the organ from the capsular and subcapsular area, okay? So all the lymph enters this lymph node from capsular and subcapsular lymphatics vessels. And when we look at the parenchyma of the organ, you will see, what is this? You will see a germinal center. This is the germinal center, and this is the cortex. And this is the cortex. Okay. lots of T and B lymphocytes. T and B lymphocytes. And B lymphocytes are localized, generally localized in the germinal center. And this lightly stained areas, this one, those ones, are all germinal center. These are all germinal center. And germinal center is surrounded by T cells, okay? T cells area by cortex. Germinal center is surrounded by a cortex. And in the cortex is stained dark here. And the cortex and the germinal center makes a lymph follicle, okay? The germinal center and the cortex which surrounds this germinal center makes the lymph follicle. So it means that the lymph node, the organ is composed of lymph follicles. These are all lymph follicles. And lymph follicles are, what are lymph follicles? Lymph follicles are germinal center, which includes, consists lots of B cells, and the cortex, which consists mainly T lymphocytes, okay? So, if we look at the medullary region of the lymph node, so this was the cortex. This was the cortex of the lymph node, and the cortex of the lymph node is made up of lymph follicles, okay? The cortex of the lymph node made up of lymph follicles, and this is the medulla. This is the medulla of the lymph follicle, and in the medulla of the lymph follicle, you will see medullary cords, which means that lots of macrophages, dendritic cells, some T and B lymphocytes as well, and there are lots of blood vessels like sinuses as well occurs in this medullary region. So in the cortex we have lymph follicles. The lymph follicles have germinal center and the outside cortex, and the medulla region of the lymph node consists medullary cords, and which are supported by blood vessels. Okay, the third organ I'm going to show you is the spleen. By the way, thymus, the very first organ I showed you, was the primary lymphoreticular organ. As you know, lymph node and spleen are the secondary lymphatic organs. Okay, spleen. Spleen is organized as white pulp and red pulp. Okay, spleen is organized as white pulp and red pulp. When we talk about white pulp, the white pulp is the made up from lymph follicles. Okay, so lymph follicles is called white pulp in the spleen. These darkly stained areas. These areas are white pulp. See, these areas are all white pulp, and all the other area dispersed in between white pulp is called red pulp. I'll try to find a better area for you. Can't see it clearly. Okay, I'll try to hold. So these are all white pulps. This is a white pulp, another white pulp, another white pulp. Okay, these are all white pulps. And in reality, these white pulps are lymph follicles. Lymph follicles. You know, in the previous slide, we were talking about lymph nodes, and those lymph nodes has lymph follicles, and this spleen also have lymph follicle, which are organized as or named as white pulp. This white pulp also have a germinal center, which consists mainly B lymphocytes, and the cortex, the area which surrounds the germinal center, which consists mainly B lymphocytes. And as you can see here, see in the white pulp, you can see arteriole. Okay, let's try to magnify our image. See, this is an arteriole, and better section. Okay. So this is my white pulp. This is one white pulp, and all the other areas in between white pulps are red pulps. And this white pulp has a germinal center inside, germinal center, which consists mainly B cells. And then this germinal center is covered or surrounded by the cortex or the T cell area. And in this white pulp, we have an arteriole, this arteriole, and the cells surrounded by this arteriole are called PALS, periarterial lymphatic sheath. Periarterial lymphatic sheath. Okay, so the cells surrounds this arteriole in the white pulp are called periarterial lymphatic sheath. Periarterial lymphatic sheath, which are mainly T lymphocytes. Okay. And in the red pulp, you will see lots of spleen cords. These spleen cords have macrophages, dendritic cells, lymphocytes again, and lots of sinusoids. Like these white openings are sinusoids. And remember, what are sinusoids? Sinusoids are capillaries whose basement membrane is intermittent, it is not complete. And remember, erythrocytes, all erythrocytes are destroyed in the spleen, in the red pulp of the spleen. Okay. And after this, in your lecture, you also have learned mucosa-associated lymphatic organs, mucosa-associated lymphatic organs, because all our body, these lymphatic organs are can be found all over our body, and these are associated with our mucosa. In this example, I'm showing you an ileum section from our gut, and this lymph node, this lymph follicle is found in the ileum. So it is related, it is gut-related lymphatic organ. It is a gut-related lymphatic organ. So this is the epithelium. Alright. You may recognize this slide. So this is my ileum, the gut section, and this is the epithelium, okay? And these are all goblet cells. Remember goblet cells, which secretes mucus, okay? And this is my gut epithelium, and beneath the epithelium, I have my connective tissue. I have my loose connective tissue. And these structures are called villi. You will learn in the next committee, I guess. And when you look at the mucosa, so are also lymph follicles. These lymph follicles also consist macrophages, lymphocytes, dendritic cells, okay? So these are another lymph follicles, which is found in the mucosa of the intestine. That's why we call them gut-associated lymphatic organs, okay? Gut-associated or mucosa-associated lymphatic tissues. Mucosa-associated lymphatic tissues, but in this particular case, this is gut-associated lymphatic tissue. And these lymph, it's important, you may hear it in your lecture, these lymph follicles are in particular called Peyer's patch. Peyer's patch, okay? These lymph follicles in the ileum, or in the gut are called Peyer's patch. Peyer's patch. Okay, that's all you have to see in this laboratory. Please try to see all these structures